In this video, we are going to discuss the workings of sex determination. You will learn the difference between sex and gender, sex determination, the different systems by which it works, and, finally, some of the disorders associated with sex determination. To start, we should define sex determination. Sex determination is the specification of sex, male or female. Generally, males produce smaller gametes, sperm, and females produce larger gametes, eggs. The male gametes are motile and are produced in great number, and seek out the less numerous but large and immobile female gametes. Sex-determining mechanisms include chromosomal, genic, and environmental sex-determining systems. It is important to note that there is a difference between sex and gender. Gender is not the same as sex. Biological sex refers to the anatomical and physiological phenotype of an individual. Gender is a category assigned by the individual or others based on behavior and cultural practices. One's gender does not need to coincide with one's biological sex. There are many ways in which sex differences arise. In some species, both sexes are present in the same organism, a condition termed hermaphroditism. Organisms that bear both male and female reproductive structures are said to be monoecious. Species in which an individual organism has either male or female reproductive structures, such as humans, are said to be dioecious. The difference between sexes is often seen through which chromosomes are present, displayed uniformly in every cell. For example, if the organism possesses two of the same gametes, they are said to be homogametic. If the organism produces two gametes that are different, they are considered heterogametic.
Now we are going to discuss the different sex determination systems seen in organisms. The XX, XY, XX, XO, and ZZ, ZW are all chromosomal sex determination systems. In the XX, XY sex determination system, males are XY, the heterogametic sex, and females are XX, the homogametic sex. The chromosomes contain pseudoautosomal regions, which are small regions of the X and Y chromosomes that contain homologous gene sequences, allowing for pairing and alignment of X and Y chromosomes during cell division. The male sperm is what determines the sex of the offspring, as it can contribute an X or Y chromosome. The Y chromosome, additionally, has many unique features. It is acrocentric and not actually Y-shaped. Additionally, maleness is primarily determined by the presence of the SRY gene on the Y chromosome. This system is prevalent in humans. Another chromosomal sex determination system is the XX-XO system. This is the simplest mechanism of sex determination. In this system, females are XX, the homogametic sex, and males are XO, the heterogametic sex. It is important to note here that there is no O chromosome. This signifies the absence of a chromosome. The sex of the offspring is determined by which type of sperm from the male fertilizes the egg. This system is present in some grasshoppers and other insects. The final chromosomal sex determination system we will be discussing is the ZZ-ZW sex determination system. One should be careful to not associate the letters Z and W with the shape of the chromosome. Rather, this is meant to not confuse it with that of the XXXY system. In this system, females are ZW, the heterogametic sex, and males are ZZ, the homogametic sex. When determining sex, the males can only contribute a Z chromosome, whereas females can contribute a Z or a W chromosome, allowing their gametes to determine the sex of the offspring. This system is present in butterflies, birds, and some reptiles and amphibians.
Other sex determination systems that are not as common but are so prevalent are genic and environmental sex determination. Like the aforementioned examples, genic sex determination is a type of sex determination in which a sexual phenotype is specified by genes at one or more loci, but there are no obvious differences in the autosomal chromosomes of males and females. In some instances, there are no sex chromosomes present that would specify sex. This system is present in some plants, fungi, protozoans, and fishes. Environmental sex determination is when the sex is determined fully or in part by environmental factors, especially temperature. This is present in some invertebrates, turtles, and alligators. An example of environmental sex determination is sequential hermaphroditism, the phenomenon in which the sex of an individual organism changes in the course of its lifetime. For example, the organism is male at one age or developmental stage, and female at a different age or stage. This type of behavior is exhibited in organisms like slipper limpets, in which the first larva to settle on a substrate develops into a female, and thus attracts other larvae. These new larvae settle on top and develop into males to act as mates for the females. After a period of time, the mates develop into females and attract new larvae to settle on top and develop into males.
The cells of human females normally have two chromosomes, and the cells of males have one X chromosome and one Y chromosome. However, sometimes there are disparities between the number of sex chromosomes an individual possesses caused by problems in meiosis. These abnormalities are expressed through different genotypes and phenotypes of the individual. There are many syndromes that can arise if there's a mutation in the sex chromosomes. Turner syndrome results when an individual is XO and has no Y chromosome or additional X chromosome to replace the absent Y. The O in XO indicates the absence of a sex chromosome. People who have Turner syndrome are phenotypically female and often have underdeveloped secondary sex characteristics. Affected women are frequently short and have a low hairline, broad chest, and folds of skin on their neck. Most women with Turner syndrome are sterile and usually have normal intelligence. Turner syndrome is not as rare as the other sex syndromes, with an incidence rate of 1 in 3,000 female births. Klinefelter syndrome is another syndrome that results from a mutation in one sex chromosomes. It occurs when an individual has two or more X chromosomes and at least one Y chromosome. Individuals with this syndrome are typically XXY. People with Klinefelter syndrome are phenotypically male and often have small testes and reduced facial and pubic hair. Most men with Klinefelter syndrome are sterile, taller than normal, and exhibit signs of normal intelligence. Klinefelter syndrome is less common than Turner syndrome, with an incidence rate of 1 in 1,000 male births. Finally, triple X syndrome occurs in poly-X females, females who have 3 X chromosomes. Females with this syndrome have a tendency to be tall and thin, and while a few are sterile, many menstruate regularly and are therefore fertile. There is a greater incidence of intellectual disability in poly-X females, but many have normal intelligence. It should be noted that females with more than 3 X chromosomes are rarer, because as the number of X chromosomes increases, the severity of physical and mental disabilities also increases. Triple X syndrome has an incidence rate of 1 in 1,000 female births.
In summary, sex determination is the specification of sex. Gender is not the same as sex. One sex-determining mechanism is chromosomal. This is divided into XX, XY, XX, XO, and ZZ, ZW. Other types of sex determination are genic and environmental sex determining systems. Many syndromes are associated with improper pairing of sex chromosomes, such as Turner syndrome, Klinefelter syndrome, and triple X syndrome.